Good morning, everyone. This is John Heerdink. I'm the managing member at Tribe Public. Many of you, I'm sure, recognize me by now, but for those of you that joined us over the last couple of weeks to get in front of the companies you care about, and specifically our presenters today, INmune Bio, a NASDAQ traded company under the symbol I, N as in Nancy, Mary, M as in Mary, and boy, B as in boy, INMB. I really appreciate all of you coming in here and expressing your interest and joining me to learn more about the companies and the subjects we care about. Today, we're going to be touching on a subject again, Alzheimer's, and the great work that this company is pushing forward, and some of the research that results that were quite amazing that have come back recently. And we're very excited. Those who remember, we had the INmune Bio, at least we had Dr. Tesi, RJ Tesi, in view there on in May, and the stock was around nine and change, if you will. And we went on a nice little run. We've gone over $30 twice, a triple here, and it's reverted back to around the $20 mark, still more than a double. And the company's been able to accomplish a great deal. I was just talking with the other man that I'd like to introduce you to, David Moss, the CFO of INmune Bio, and at the amazing pace and amount of accomplishments they've been able to bring forth over the summer is truly amazing. I commend you guys, and I've been tracking the company, investing in the company over the last couple of years through my Vista Partners investment arm. Truly impressed with what they've been able to do and their commitment to moving this company forward. So Dr. Tesi, if you wouldn't mind, just a quick couple words about your background, and David the same, and then we can go on and with our presentation after that.
Yeah, thank you, John. So I am a founder. The company was started in 2015, but actually both of our main platforms, although we're only talking about DN-TNF today, our XPro platform, I've worked with for more than a decade. I've started working with XPro in 2011. So I had 5 or 6 years with the drug before we actually formed the company. I have been in biotech for the past 20 plus years, but before that, I was an academic transplant surgeon. So I'm one of the lucky few that have been on both sides, so to speak. I've taken care of patients with cutting edge technology, and now I'm in the process of doing drug development for cutting edge technology, both treating unmet needs. And in fact, that's what gets me up in the morning. I look at a problem that has not been solved by modern medicine, and I say, that's what I want to do, and then I go do it. David?
Yeah, no, thanks, John, and certainly thank you for the audience for allowing us to present to you. I've been biotech for almost all of my career, 25 years. Started in San Diego, ended up in the medical center at the Houston Medical Center in tech transfer, helped start companies, ended in the venture community, and really have done everything from putting the science together, putting the boards together, the initial founding round, the seed round, the Series A, B, C, Ds, the bankruptcy, shutdowns, IPOs, mergers, acquisitions, partnerships, essentially a little bit of all of it. So I think one of my strengths is really trying to bring all the right pieces together at the right time to help build a company. And I think the fortunate thing about RJ, myself, and Mark Liddell, who's not on this call, is that we're all, number one, we get along like a house on fire. We're really friends first. And, we really appreciate our shareholders. We are investors ourselves, number one, so we know what investors want because it's what we would want. And I think good companies are built when investors think like management, and most importantly, management thinks like investors. We're very large shareholders. We're large investors ourselves, not just in this company, but we invest in other companies as well. And we try and operate our company in a very simplistic form. So maybe with that being said, John, if it's okay, I'm just going to jump into the presentation.
Yeah, just one additional reminder is to all the tribe, feel free to begin sending additional questions. Some of you already sent them over, but we want to get the questions that you want answered. So just send it through the chat feature, and we'll do our best to address those and move forward. But please continue to do that as we go through the Zoom chat feature. Thanks.
Great. Thanks, John. So forward-looking statements, you're all familiar with it. Back to thinking like investors, one of the things we like to think about is the simplicity of our capital structure. We often find that complex capital structures are difficult to understand. And frankly, if you don't understand them, you shouldn't invest in it. So why even dig into the science if the cap structure is too complex? We've never sold anything but common stock, even as a private entity. It made things a little bit challenging when we did that. But as a result today, there's never been a warrant, there's never been a preferred, a convertible debt, or any complexity. It's kind of what you see is what you get. There's about 17.8 million shares of common outstanding. Over 30% is owned by the insiders. And it's easy to figure out what our market cap is. You look at the share price, you look at the number of shares outstanding, and you can figure it out. You don't have to figure about a resetting warrant or a preferred, et cetera. As many of you know, we focus on dysfunction of the innate immune system. We do have two platforms that modulate and repair dysfunction of the innate immune system, which make a difference across a wide variety of different therapeutic indications. Today, we're only going to focus on one, which is the dominant negative TNF platform, or DN-TNF. Don't need to worry about what that gobbledygook means, but I'll say that the dominant negative technology is the first of its kind in the clinic. And this dominant negative technology allows us to do things that are well beyond what the existing drug franchise, which are called TNF inhibitors, are allowed to do. That drug franchise are drugs like Humira, Remicade, and Enbrel. These are drugs you've probably heard of. They sell north of 40 billion a year, and they're really trapped in the autoimmune disease category. That's diseases like rheumatoid arthritis, psoriasis, Crohn's. And using this dominant negative technology, we're able to go into areas like neurodegeneration, infectious disease, cancer, et cetera. And what we're going to share, what RJ is going to share with you a little later today, are some of the discoveries, some pretty remarkable discoveries that we've made using this drug in our clinical trials. We're very, very excited about this data, and I think it's quite novel, and it's something that we're very, very, very passionate about. The second platform is around natural killer cells. We won't talk about that today, given the time, but that's in the clinic. And there's some interesting data that's being generated. And maybe one day, John, we'll have a chat just around natural killer cells.
Love that. Thanks.
Yeah. In the meantime, there's more than 70 publications. These are not sponsored by the company. These are publications from universities around the world. They came to us because they saw the application of the drug, and they felt that there was, they knew that there was an application that would help them with their scientific experiments. That's what resulted in close to 70 publications. It gives us a lot of foundational support in the work that we're doing. And on top of that, we received now close to $5 million in non-dilutive funding from agencies such as the NIH and the Alzheimer's Association to move these programs forward. This is what we were talking about a little bit earlier, that innate immune dysfunction can drive a lot of different disease states. More than 50% of all attributable deaths are related to some inflammatory condition. And it's really about being able to target inflammation, but it's as important in how you target this inflammation. And I really want to emphasize the how you target. And this dominant negative technology that RJ will speak about is the how. And by doing so, we believe we can cut across almost all of these diseases you see listed here. In fact, all of them that you see listed here, we can make a difference. We're starting in CNS disease, in neurologic disease. And we have run a phase 1 trial in Alzheimer's, and you will see that data here shortly. So RJ, I'm going to pass this to you and let you talk about how we treat Alzheimer's as an immunologic disease.
Yeah, thank you very much, David. And, my goal is to really help you understand why we're excited about what we're doing and justify that based on the data we have at hand, and tell you about the promise that's on the horizon. I think one of the things that we do differently is we treat Alzheimer's, or view Alzheimer's, as an immunologic disease. People kind of scratch their head and say, but isn't Alzheimer's a disease of the brain, of the central nervous system? Yeah, it is. But it's really, that's just because that's where it's occurring. We don't think as approaching it as a neurologic disease has been that successful. I mean, you see all the controversy around the Biogen drug and all the problems that have been, the challenges, I guess a better way to say it, of developing drugs for Alzheimer's and CNS drugs in general. But targeting it as an immunologic disease really changes the way we approach it, gives us lots of opportunities, and I'll tell you about those now. Next slide. So, we think most people make Alzheimer's disease way too complicated. One of the things I've learned in medicine and drug development is usually the simple solution is the best solution. As soon as you get too many moving parts, it's just very hard to make things work. And when you really strip away everything that's all about Alzheimer's, what's happening? You're having nerve cell death in the brain. That's the memory bank. That's where the memories are stored. That's where your daughter's birthday is. And then you have synaptic dysfunction. Synapses are the wires that connect those cells, those bits of memory. And when those two work together, you can remember your daughter's birthday, right? When one or the other get messed up, you can't find your daughter's birthday and you can't recall it. And that's called cognitive dysfunction. Now we're focused on the microglial cell there. The microglial cell is the immune cell of the brain. It's about 10% of the cells of the brain. And the astroglial cell, it's another immunologically active cell. It's often called the nursemaid cell that is important for the care and feeding of nerve cells and synapses. And that's 40% of the brain. So 50% of the cells in the brain are immunologically active. So of course, this is an immunologic disease. You notice I haven't said anything about amyloid and tau because we think amyloid and tau are important, but they're really kind of two-bit players here. They're on the sideline. They're in the playing field. They're in the stadium, I guess, a better way to put it. But they're not between the goalposts. They're sitting on the bench. So we think that actually targeting neuroinflammation and some of the other new therapies out there are going to be a lot more important. So let me give you a brief overview of where we are today. We have completed our phase 1 trial, and quite frankly, we've hit it out of the park. And what do I mean by that? A phase 1 trial, which is the first time you use a drug in man in a given disease, should do 4 things. The first thing it should do, it should show that it's safe in the target patient population. In this case, that target patient population is patients with Alzheimer's disease. By definition, they are patients who are 70 years old or more. Number 2, you have to have target engagement. In other words, you have to show that the drug is engaging that piece of biology that you want it to, right? I mean, it's great if it does it in a rat, but a rat has 4 legs and man has 2 legs. The third thing it needs to do, a lot of people don't think about this, is it's great if you can show that there are downstream benefits to that target engagement. That gives me as a drug developer, confidence that, when I move into phase 2, I have a good chance of success. And the fourth thing the trial needs to do is help you design the phase 2 trial so it works. Now, this phase 1 trial did all of those. It certainly proved it's safe. It's a drug that we've had patients on the drug for a year. It decreases neuroinflammation. That was the primary goal. Neuroinflammation is target engagement. And as you can see in the first 2 bullets, we decrease neuroinflammation, whether you measure it by looking at inflammatory cytokines in the CSF, which is fluid that you get from around the spine, which is a direct reflection of what's going on in the brain. And that decreases at 15% by 3 months. And it decreases by almost half if you follow those patients out for a year and you use non-invasive measures of neuroinflammation. And we'll talk more about that in a moment. So clearly we're safe. We engage the target. Now, what about those downstream benefits? Well, remember, I focused on nerve cell death and synaptic function. Those are the 2 things that if your drug isn't doing it in Alzheimer's disease, you're not in the game. We are in the game. And as you can see, we decrease neurodegeneration. That is, we improve nerve cell survival by decreasing neurofilament light chain by 84%, by decreasing phospho-tau 217, the most sensitive biomarker of Alzheimer's disease, and decreasing VILIP-1. So these are big decreases, and this is at 3 months in the CSF. We also, the last 2 bullets, improve synaptic function. Synaptic function is, that's allowing the nerves to talk to each other. Remember, there's 2 things going on here. You've got the memories are stored in the cells, and you got the wires connecting the cells, and both have to work. If the wires are cut or not talk, or the cells can't talk to each other, if the wires are there but the cells are dead, you're not going to recover your daughter's birthday. Okay. So we improve synaptic function dramatically. Now, what about these neural repair biomarkers? These are pretty interesting because these are non-invasive biomarkers that we think are on the cusp of revolutionizing CNS drug development, including Alzheimer's disease, because, and it's a topic of the presentation today, because these can very accurately measure white matter pathology. Next slide. Now, what is white matter pathology? Well, traditionally we talk about gray matter and white matter. There's the slang there that somebody's smart. Well, they got a lot of gray matter, right? Nobody says that person's got a lot of white matter because nobody cares about white matter, right? Neurologists haven't cared about white matter, except when you're talking about patients with MS, multiple sclerosis. Nobody cares about white matter because white matter aren't where the nerve cells, where the memories reside. The nerve cells that contain your daughter's birthday is in the gray matter. But it turns out that gray matter is useless if the wires aren't connecting everything together. It turns out that the white matter contains these myelinated axons. Axons are the wires that allow nerves to talk to each other. Myelin is like the insulating sheath on that wire. And these things are put together in bundles called white matter tracts, which are the superhighways that allow parts of the brain to communicate very well together. So, for instance, in language and short-term memory, there's something called the arcuate fasciculus, which connect Broca's and Wernicke's area. You don't need to remember all that. But the fact is the brain is organized in a very special way. Nobody's really thought about white matter pathology because it's very hard to study. You can't do biopsies on brains in living patients, right? Nobody's going to do a brain biopsy on me, I can tell you that. You can do it in cancer, you can do it in liver disease, you can even do it in heart disease, but you can't do brain biopsies. So neurologists and drug developers have been running blind until recently. Now with these modern MRI techniques, we can look into the brain and we can actually see what the hell's going on in the white matter. Now we can actually look at pathology in the live brain that allows us to pinpoint the location of that pathology, quantify it, and most importantly for what we're talking about today, we can actually show that when you give XPro, that pathology gets better. And what we'll show in the phase 2 trial is that correlates with an improvement in cognitive function. So let's talk about the primary endpoint. So let's talk about shall we say, the gold standard. The gold standard, I said, was you put a needle in someone's spine, you draw some of that fluid, CSF fluid, and you measure it using, in this case, we use the Olink 48 cytokine panel, and you can see it on the bottom. All those blue lines mean the cytokines are going down. Now, some people go, well, what's the most important one? And I say, that's a kind of a fool's game to say that this cytokine is more important than that cytokine, because the immune system is exactly that. It's a system. It's this complex interplay between many cells. Things are going up and down, but in general, you can see there's a dramatic decrease in the inflammatory cytokines in the spine. And many of these are from the innate immune system, which have been ignored historically. So that's invasive. That requires you to stick a needle in someone's spine. I don't know if anyone's had an LP, but it's a little bit scary. Doctors don't like to do it. It takes time. Patients are nervous about it. But what about doing it non-invasively with an MRI scan? Well, we can measure neuroinflammation in the white matter using something called white matter-free water, and that's the top figure. And you can see in the blue line that over a 12-month period, we decreased by almost half the amount of neuroinflammation in these patients with Alzheimer's disease. Now, we didn't have a control group, but we used a contemporaneous control group called ADNI, which is Alzheimer's disease neurology imaging database. And as you can see over time in the gray line, patients with Alzheimer's disease, their neuroinflammation gets worse. So that difference is dramatic. Look at 12 months. It's a 50%, it's a 55% difference in the patients who have Alzheimer's versus don't. This is a big deal. This is our primary endpoint. But now it's only beginning to get interesting. On the next slide, you can see that if you look at the, no, go back one, David. If you can look at the elements, the other two parts of what we call the virtual biopsy, that you see dramatic improvements. On the left, you see apparent fiber density, which is a measure of those axons, those wires in the white matter tracts connecting parts of the brain that work together. In a normal patient, in the gray line, though, the number of wires declines over 12 months. In our patients, they went up dramatically, 15%. People have never seen this before. They've never seen it for two reasons, because first of all, not a lot of people are using the MRI to look at the white matter pathology. The second thing, they've never had a drug that's done this, right? XPro improves apparent fiber density, and it also improves myelin. Myelin, remember, is that insulating fat that goes around the wire in the white matter bundle. Just think of a bunch of copper wires. If you got a bunch of copper wires that don't have insulation, they're going to short out. They're not going to work very well. So you need that insulation. And the problem is patients with Alzheimer's disease do demyelinate. As you can see on the right-hand figure, that gray line, the myelin, that increasing line means demyelination. If you treat them with XPro, you get a rapid remyelination when you get rid of neuroinflammation. So now we have the holy grail, or excuse me, the holy trinity of white matter analytics, where we can see inflammation, we can see axonal fiber function, and remyelination. That is completely novel. The final thing here in this figure is I mentioned the importance of synaptic function. Unfortunately, we can't yet measure this by non-invasively. You have to stick that needle in the back. I predict we'll be there soon, non-invasively, but they're not there yet. And once again, XPro dramatically increases synaptic function. So we are in the position where we have all this great information, and we're still stuck with a stock value that is less than our peers. And why is that? Because we're using these very new biomarkers that a lot of people, even people, neurologists in the field, don't understand very well. So the thing we've done recently is kind of connect them to stuff that people know. As you know, a lot of people get excited about amyloid and tau in Alzheimer's disease. Well, if you can see on the left-hand figure, we dramatically decreased phospho-tau 217 after 3 months. Phospho-tau is the most sensitive biomarker of neurodegeneration in patients with Alzheimer's disease. So doggone it, we are helping cells survive, decrease death. The second thing people like to do is they say, oh, in patients with Alzheimer's disease, the brain shrinks. If you have an effective therapy, the brain's not going to shrink fast enough. Well, I think that's a pretty lousy goal. I want my brain to shrink slower. I don't want my brain to shrink. I want it to go back to normal. And that's what happens when you get XPro. You actually show an increase in white matter volume over 12 months. No one's ever seen this before because no one's had a drug. Now you're going, well, he's pretty excited about this, but he hasn't talked about cognition. And you're right, I haven't talked about cognition because our phase 1 trial was not designed to measure cognition. It was designed to demonstrate safety, designed to demonstrate target engagement, which is decrease in neuroinflammation, designed to show improvements in benefits of downstream's effective target engagement. That's less nerve cell death and improved synaptic function. And we knocked it out of the park. We were not powered, nor did we have a placebo group that allowed us to really make comments that are meaningful about cognition. I can give you some anecdotes. We had one guy that went back to work. We had another patient who, basically went from not interacting with family to interacting with family. So we have anecdotes, but quite honestly, we don't hype those, because the only way you have a clinical benefit is with a blinded randomized placebo-controlled trial. And that's exactly what our phase 2 trial is. Our phase 2 trial, on the next slide, shows that is a small trial, 200 patients, of which there's a 2-to-1 randomization. That means of those 200 patients, 2 out of every 3 get drug and 1 gets placebo. That's an enrollment strategy. Patients they don't like to think they've got a 50-50 chance of drug. They'd like to think they have a 65% chance of getting drug. And it's only 6 months. Now think about that, 6 months. Every other Alzheimer's trial, 18 months. Ours is a lot shorter. How can we do that? We can do that because we don't take all comers. We take patients that we think we have neuroinflammation. You see those 4 criteria in the left lower corner: elevated CRP, SED rate, hemoglobin A1C, or the ApoE4 gene expression. Those biomarkers suggest someone has neuroinflammation. About 50% of patients with Alzheimer's disease are eligible for our trial. Now, why do we do that? Well, we did this because we learned from cancer trials. The oncologists get more drugs approved in a year than CNS drug developers do in a decade. And why is that? Is because they use biomarkers, they match patients with the drug and the disease, and they're successful. CNS has not done that. We want to change that. The other thing we've done is our primary endpoint, which is a cognitive endpoint, is something called EMAC. It's not ADAS-Cogs 13 or 11 or whatever people are using. And why is that? It's because if you're treating patients with early AD, ADAS-Cogs is just not sensitive enough. In an ADAS-Cogs 13, 9 of the 13 elements have reached a ceiling effect in patients with Alzheimer's disease, mild Alzheimer's disease. That means, in fact, it's an ADAS-Cog 4. It's so insensitive that you certainly can't see if someone has stable disease. You certainly can't say if someone has improvement. And you're lucky if you can tell if someone, how fast they're getting worse, and whether you're not getting worse as fast as your placebo group. Now, if someone's goal in drug development is to decrease the rate of getting worse, I'm in the wrong business. I went into medicine to make people better. And that's exactly how we're doing our trial. Our goal is the day a patient starts on his drug, he won't get a bit worse. His cognitive function will stay the same. That is our goal. We think if he gets worse, that's not very good. In fact, we hope they get better. I'm not going to promise you that. I got my fingers crossed, but I'm not going to promise you that. We think everybody is going after this disease with not a very ambitious approach. Now, based on our phase 2 trial, we think this ambitious approach is worthwhile. Mm-hmm. So in summary, because we're at 59, getting close to the end here. What's the last slide, David? I can't remember.
You're going into that's it, really. You're going into the MOA after that. We can use that if there's a question.
So the bottom line is that we have done a very successful phase 1 trial. We know everything about the biology. We know what patients we want in our phase 2 clinical trial. We've designed that clinical trial. We have the right dose. We have the right duration. We have the right biomarkers for inclusion. And we believe that we have the perfect drug for the perfect group of patients to get the kind of result that we want, that patients want, and what investors want, which is going to be improvement in cognition. So I'll stop there and take questions.
Yeah. Thank you, RJ. And thank you, David. Appreciate it. Reminder to all the tribe, please just send your questions to the Zoom chat feature. and I'll try to get those addressed time permitting. We'll look to get INmune Bio back on here soon. So if you don't get it, don't fret. We'll get them back on. It's too meaningful of a topic to not have this out in front of the tribe, in my opinion, and many of you expressed that too. One of the questions that came up, one of these stories. People love stories, and maybe it's just a story, and you're not comfortable to talk about. But I believe when I spoke to, it was either you or David, sometime over the course of the last couple of years, you spoke about I think an educator that had to retire that then reverted back to work. Is that
so? That's our star patient. Of course, we love this patient. So he was a principal of a boys' high school, and he had to retire because he stopped remembering names, had to forget, and he was relatively senior. I think he was 68, 69, and he just couldn't do his job anymore. And he was just completely depressed by that. He got in the trial. And by 6 months, by his belief, by his colleagues' belief, by his clinical team's belief, he was completely back to work, normal, and he went back to work. Now, to me, that's a rip-roaring success, but I want to emphasize it's an anecdote. It's a story. It's a story that I think about every day, because that's what I want our drug to do for everyone with mild Alzheimer's disease. To be clear, if someone has very, very severe disease, they're wheelchair bound, they can't feed themselves, I don't know if XPro is going to make a difference in their life. I would doubt it would be. But more than 50% of patients with Alzheimer's disease have mild enough disease that I think if you fix axonal degeneration and then repair synaptic function, you're going to make a difference in their lives. And remember, when you're treating patients with Alzheimer's disease, you're not just treating the patient, you're treating the family, you're treating the caregiver. It is a unit. If any of you have had the misfortune of having someone develop Alzheimer's disease in your family, it is a very difficult scenario. Someone has to basically become responsible for that patient's life, whether it be physically or financially. They need help. We want to change that.
Agreed. Unfortunately, it has touched way too close to my home, and I saw it, take down to a degree, a massive 10-person family that had to take care of, their mother, and it just over a 10-year period, and it was heart-wrenching. And so I'm so excited about the work you're doing and that you're bringing to the table, and wishing and praying and everything else that you guys bring it to the market as soon as possible. One of the switching to the financial sides of things, you've had a couple raises this year that have really substantially boosted your financial position, and you've done it very conservatively, in my opinion. As you mentioned earlier, it was straight common that you issued 2 raises. One, I believe, in January of this year was at $20. The second one was at $22 over the summer. After the stock, ran to 30, you guys were able to capture. Could you speak to that, David? Exactly where you are per what's in the public domain now, where financially, and where that takes you?
Yeah, we'll be reporting our quarter soon, but you can rest assured that basically from the last quarter, which we reported just under 40 million, and then we raised about 55, that would put us, somewhere in the just close to the $90 million range. Really, our goal was to get enough money to fully run the entire phase 2 program, take it all the way through, and that's where we are today. We've got money well into 2023. It doesn't include anything that we try and do on the non-dilutive front, which we have always tried to do. And as a reminder, we do run some of these programs over in Australia and the UK, and we do get rebates from those governments for what we spend over there. Mm-hmm. So we continue to do that as well and run as efficient a capital portfolio as we can. The rebates that we get back, we reinvest back into the clinical programs. And, we run things very simply, right? We do have a bit of debt that we took on from Silicon Valley Bank. We did that to buy back basically an option that Xencor held. So we really reduced our future capital structure by buying back stock. And that's just not seen in many biotech companies. How many small biotech companies that buy back their own stock, right? So we constantly monitor the situation, and we try and be creative when we can be creative is really what we do. But we're sensitive as much as we can be about how we raise money. We've so far to date, we've only done up rounds. We want to maintain that that record, and we are being large shareholders. We're just sensitive, like we said from the beginning. We're investors too. And we know investors never like it when you dilute companies, and it's really about how you do it and when you do it, but we're sensitive to it. It has to happen in biotech. Biotech is an expensive business. We need money to move our trials forward, and we always look at the cost benefit of doing it.
Thank you. This question just came up from one of our friends, Rob, over in Switzerland, one of the family offices. He's saying, has the FDA been generally on our side, in quotation marks, in terms of approving CNS therapies and AD specifically? And his second question is AD an area of focus for the FDA currently? How would you characterize this?
Yeah, so I think, clearly the fact that they allowed Biogen to reach the market means that they now understand that Alzheimer's disease is a desperate problem that needs to be solved. So I believe they are on our side. Now, that doesn't mean they're going to, just keep the door open and let everyone pour through. You have to meet some minimum criteria. And I think they're going to be conscious of that. And that is, you have to be safe, you have to show efficacy, and ideally you have a biomarker that correlates somewhat with your drug response. Those are the three criteria. So I am a big believer that there are going to be more Alzheimer drugs approved in the near future. I mean, the CEO of Lilly, which has donanemab, which is an anti-amyloid drug, has made the statement that they're going to file their phase 2 data by the end of this year, 2021. Now, admittedly, they have a 1,500 patient phase 3 ongoing, so it's not like they're trying to just get approved with a phase 2. But the fact is, the FDA has opened the door to fast track approval strategies, where traditionally CNS is only used kind of the slow track approval strategies. So we think that the FDA is going to be very supportive of what we do. But I want to make an important point. This is not just an Alzheimer's drug. If you're got an anti-amyloid drug, the only thing you can treat is Alzheimer's. If you have an anti-tau drug, you can treat Alzheimer's and FTD, and that's just about it. We target neuroinflammation. Neuroinflammation is present across the board in CNS diseases, whether you're talking about neurodegenerative diseases, MS, Alzheimer's, Parkinson's disease, Huntington's disease, ALS, whether you're talking about psychiatric diseases, such as depression, bipolar disease, PTSD, whether you're talking about acute neurologic injuries, such as traumatic brain injury or stroke. Neuroinflammation is involved in all of those. So the Alzheimer's program is just the first step with this drug into many markets that have unmet needs. And everything we learn and use, every trick, every biomarker we use in this Alzheimer's program to develop this drug for Alzheimer's, we'll be able to apply what we learn to those other diseases. We've announced our treatment-resistant depression program, but I can tell you the list is long. And if you want any idea how long that list is, go to our website and you'll see in those 70 publications, there's a lot of different neurologic diseases that have been cured in mice.
Thank you. The switching gears, going back to the list of questions we have, one is in regards to again the stock side of this scenario. Could you confirm how many analysts are currently covering you? And I believe the highest price target right now is $42. Can you speak to that, David, and any comps that you might give the tribe to sort of look at as well would be appreciated.
So I think there's B. Riley, Maxim, and BTIG that are covering us.
Mhm.
And we're working to obviously increase that notoriety. With regards to, I'm sorry, the second part of your question, John?
Well, there's one is the price target. Where do they range currently with those analysts? And I believe
and the proxies, yep. So, and also Roth, by the way, sorry, and Roth. So those are the four. I can't comment on how they come up with their price targets. I think what they're really doing, the problem we have with our analysts is that we're both a CNS company and an oncology company with two different platforms. When you go to speak to an analyst, you speak to either somebody who's an expert in CNS or an expert in oncology. You never find an analyst that's an expert in both because of their training. So it doesn't really fairly value, I think, both of the platforms. My guess is that the plugins that they're using in their models is just one or the other, not both. So in the case of the Alzheimer's disease, the numbers can get out of control really quickly, but they're probably modeling some patient population and some price and some percentage of success, some success rate. Sure. But they're not modeling INKmune, and then the other way around, right? INKmune's just really come out of the gate. It's new. And when you come to proxies, there's really two buckets of proxies that I think you have to add together for us. There's the proxies that you have in CNS disease, and then there's the proxies that you have in the NK space. In the NK space, I think the proxies are pretty easy to find because there's not many companies. Those are obviously companies that are really working on pure NK cell related platforms. Those are companies like Nkarta, Fate, and Century and others. And then if you look at the CNS area, those are Elector, Denali, and on down the line, right? Those that are looking at neuroinflammation as a target. But I'll also, leveraging on what RJ said, this drug, we have data in Parkinson's, we have data in ALS, we've had some funding in ALS to set up some clinical work, and on and on. And there's other companies that are targeting those areas as well. But really, I think that what we have to do is go further down the clinical development, share more patient data, and then hopefully the market properly rewards us.
Sure. Yeah, I think the couple of reports that I have access to, the price targets in the 31 to 42 range, or in, roughly around there from B. Riley and Maxim, et cetera. So it's significantly higher than where you're trading today. And I know we were just there a heartbeat ago, and the tight float, low float nature of this with high insider ownership allows this volatility. But overall, you keep building. You've added to the balance sheet something to tune of close to 100 million or so, on my estimates, where you are. And I guess one of the last questions is, what milestones are we looking at between now and year end, and then maybe in the first half of next year? What's out in the market today?
So the thing that we believe our Alzheimer's program is really the most visible one for investors. That is what we will be held to, and our goal is very bright goal is to get those first patients enrolled before the end of the year. Now, that doesn't sound like a very big lift, but it's a huge lift. There's a lot of competition for patients out there. We expect to announce additional trials starting in 2022, and we hope to have additional patient data in the INKmune program. David mentioned the INKmune program, which is the NK program, being talked about in both this year and next year. But the bottom line is that we continue to be active. We have a great drug that is safe and effective. We have opportunities for both dilutive and non-dilutive financing, and we will make a difference at the bedside, not only in Alzheimer's disease, but in many other diseases. And that's where the value really comes. And I might add, nobody has written a rule that Alzheimer's disease has to be treated with monotherapy. That is to say that Alzheimer's is a complex disease. We expect that most patients will get combination therapy, and we expect that almost everyone will need a neuroinflammatory strategy to go with that combination therapy. For instance, we would predict that if you combined our drug with Biogen's drug, that not only would it improve the safety profile of Biogen, but it would also, of the Biogen drug, it would dramatically improve efficacy. So in fact, we think the future is bright in Alzheimer's, but we think the future is bright for XPro and CNS in general.
Well, thank you, gentlemen. I appreciate it. Thank you to all the tribe members that joined us today. Proud to have you guys on the program and look forward to having you back. And we'll get more into that NK cell platform that many of you on the tribe have been investing in for a couple of years at Fate, where we've done an amazing, seen a matching swell in our valuation. Similarly, INmune Bio's got some exciting platform as well. We need to learn more about that, but that's for another call. And I would just say, continue to send in your interest. If you want them back on, continue to add to your wish list. And note that I will get the video of this event up at the YouTube channel for Tribe as soon as possible this week. So if you want to refer back or push this out to your other friends that weren't able to make it today, it'll be available soon. Again, thank you, gentlemen, RJ and David, and INmune Bio. Remember, the ticker on INmune Bio is INMB on the NASDAQ. And I look forward to hearing from you soon. Thanks again, all, and have a great rest of the week.
Thank you.